Greetings. My name is Mikayla Rodriguez, and I am an undergraduate research student at the University of Texas, Permian Basin. Today, I will be discussing my undergraduate research project titled Zinc Oxide Nanotechnology Expenditures in the Production of Agricultural Provisions. I was able to accomplish this project with the help and guidance of my research professor and mentor, Dr. Mikayla, also receiving gracious financial support from the Welsh Foundation and undergraduate research program at UTPB. Nanotechnology refers to sophisticated technology in which materials we call nanoparticles, which range from 1 to 100 nanometers in size, are manipulated in such ways that they can carry out various functions. Some of these functions include biomedicine and drug delivery, cosmetics, textiles and fabrics, optics, energy, and in agriculture. In agriculture specifically, the most common uses of nanotechnology include controlled release of micronutrients, pesticide and fungicide properties, decreased loss of crops, and altered plant nutrition. The goal of this project was to observe the effects of a zinc oxide nanoparticle solution in comparison to a zinc nitrate fertilizer at varying concentrations. This was tested against two basil plant varieties, sweet and Persian basil. The basil seeds we used for this project were purchased from Pine Tree Garden Seeds, and the zinc oxide nanoparticles were purchased from U.S. Research Nanomaterials based out of Houston, Texas. To enable efficient seed germination, the seeds were primed by soaking them in deionized water and stirring with a magnetic stir bar for about 15 minutes. The primed seeds were then left in a dark, cool place and observed until the seeds germinated into seedlings. 8 days after seed priming, the seedlings were transplanted to soil, placing 3 seedlings per pot of soil. Subsequently, the plants were maintained at 65% relative humidity, 20 to 25 degrees Celsius, and a 16 8-hour light photo period while they matured. During this time, the fertilizer suspensions were prepared at varying concentrations for both the zinc oxide nanoparticle solution and the zinc nitrate solution. For each type of fertilizer, suspensions at 0, 0.5, 1, and 1.5 parts per million were prepared. To ensure adequate homogenization of the suspensions, they were placed in an ultrasonication bath for 1 hour at a frequency of 1200 kilohertz at room temperature. Presently, there exists multiple methods of applying nanoparticles to plants. In this particular experiment, the foliar application method was selected. Each fertilizer suspension was placed in a sterile plastic spray bottle in 10 milliliter aliquots and stored away from direct light as to avoid photoreactivity. 72 days after the seeds were transplanted to soil, the fertilizer treatments began. The plants each received 4 2.5 milliliter treatments 4 days apart. In total, each plant received 10 milliliters of a fertilizer solution in the duration of 16 days. Samples of sweet and Persian basil were treated against the 2 fertilizers at each of the 4 concentrations. The plant samples treated at concentrations higher than 0.5 parts per million appeared to experience leaf discoloration. This occurred for both the zinc oxide nanoparticle suspension and the zinc nitrate suspension. Interestingly, the discoloration occurred more frequently in the plant samples treated with zinc nitrate. Zinc nitrate can be found in most widely used fertilizer sprays. In addition, the plant samples at concentrations 1 and 1.5 parts per million seem to grow at a decreased rate than those at 0 and 0.5 parts per million. This was largely observed in the shoot region of the plants. The preliminary findings of this project offer evidence suggesting that perhaps nanofertilizers applied via the foliar route may have less harmful effects on plants than other widely used fertilizer sprays. Consequently, further exploration of nanotechnology applications in agriculture could lead to more efficient and environmentally friendly practices of growing crops. In conclusion, the findings of this project suggest that at concentrations lower than 0.5 parts per million, nanoparticle fertilizer may be more positively correlated to plant growth and development than other widely used fertilizer solutions. When treated at a concentration higher than 0.5 parts per million, the plants were observed to have discoloration of leaves, as well as a decreased rate of growth. It is important to note that the previously mentioned observations occurred less frequently in the zinc oxide nanofertilizer spray. In the upcoming months, more detailed and specific evaluation of the samples will investigate the biochemical constituents of the basil plants. This project received funding from the esteemed Welsh Foundation, as well as the undergraduate research program at UT Permian Basin. I would like to give special thanks to my research professor and mentor, Dr. Minka Montes, as well as the Department of Chemistry at UTPB for the continued support and guidance.